Yeah, hello, good afternoon, students. Welcome back to Botany second year class. So till now we have finished the SAQs and the LAQs of all the chapters. Now we will go with the VSAQs that are very, very important for us to go for the supply exams. Only for the supply exams. Okay. So in this, from the first chapter onwards, we will take out this VSAQs. The first one is what are porins? What is the role that they play during diffusion process? Porins means a small protein holes that are present on the membrane of the plastids, otherwise the cell wall, mitochondria or anything are called as the protein, called as the porins. And these porins will make the movement of the, otherwise they will allow the molecules to move from one place to another place of the different sized protein molecules, that is called as the porins. They will help out in the diffusion, that is movement of the molecules of the different sizes from one place to another place. Okay. Next one is the water potential. Define water potential. What is the value of water potential of pure water? Water potential means water potential is the chemical potential difference between the solution and the water molecules, and the pure water potential is 0. Okay, that is the one what we have to write. The next one is the differentiate between osmosis and the diffusion process. What is meant by osmosis? Movement of water molecules from the high water potential to lower water potential. That is the otherwise high chemical potential to the lower chemical potential. That is called as the osmosis. And the next one is the diffusion. Diffusion means movement of solute particles from the region of higher concentration to the region of lower concentration without having any membranous is called as the diffusion process. And this osmosis helps the plants to absorb more amount of water, and this takes place in the liquid medium only, osmosis, whereas the diffusion helps out in the gaseous exchange and it takes place in all sorts of the forms of the particle, that is the liquid, gases and the solids. Okay, next one. What are apoplast and symplast? Apoplast means what? What is the symplast? We need to define it and we'll get 2 marks over there. See, apoplast means when the water molecules will move within a plant system, within a plant cell, through the intracellular spaces or through the spaces between the cell wall and the plasma membrane is called as the apoplast. If this is the cell wall, the plasma membrane, what we will draw, and with this, otherwise through the intracellular spaces. Okay. When the water molecules will move, that is called as the apoplast. And the symplast. In symplast means when the water molecules will move from one cell to another cell through the plasmodesmatal connections. Through the protoplast and the plasmodesmata is called as the symplast. Okay. So this is from the aerial parts of the plant to the stomata, cuticle and the lenticels. Like that. Now comes the next question. What is guttation? How does guttation differ from transpiration? Transpiration means loss of excess amount of water molecules in the water vapor, in the form of water vapor through the stomatal apparatus is called as the transpiration. Okay. Transpiration means loss of water molecules in the form of water vapor through the stomata, not the cuticle lenticels. That is called as the transpiration. And guttation means loss of water in the form of liquid through hydrothores is called as the guttation. And this guttation will be along with whenever the water was lost through the guttation, that will be along with the salts and the minerals. Okay, and through the transpiration, only water is going to be lost in the plant body. Now comes the next one. What are the physical factors responsible for the ascent of sap through the xylem in the plants. So the physical factors responsible for ascent of sap are the cohesion, adhesion, and the transpirational pull. Cohesion, adhesion, and the transpirational pull. Cohesion means attraction between the two water molecules. Adhesion, attraction between the water molecules to the inner wall of the xylem and the tertiary elements. Transpirational pull, through which the water will be moving from the roots to the upper parts of the plant body. That is the one. Okay. Next question, what we will get at is the does transpiration occur at night? Give an example. In the normal plants, transpiration never occurs at night. But in some plants, that is the succulent plants. In succulent plants, transpiration occurs at the night time only. Okay. At the night time, transpiration occurs in the succulent plants, which show the scotoactive stomatal apparatus in them. Okay. So example is the bryophyllum leaf. Bryophyllum leaf. If you cannot able to remember this bryophyllum, then you can make the cactus. Easy na? Opentia, cactus. Anything you can write. And the next one. Compare imbibing capacities of pea seeds and the wheat seeds. Imbibing capacities means when the seeds are dry, when they will absorb water and become swollen up. That is called as the imbibition. Okay. Dry seeds when we soak, just like the groundnut seeds will soak, even the chana seeds will soak now. So like that. Okay. Pea seeds and the wheat seeds. So the pea seeds are the much more proteinaceous and have more imbibing capacity than the wheat seeds, which are having the starch more in them. Okay, because proteins have more imbibing capacities than the carbohydrates. Okay, pea seeds and the wheat seeds, and in this proteins are more. In this, carbohydrates are more. So, proteins will have the high imbibing capacity. And carbohydrates are having less imbibition capacity. If you remember this, if you write like this, 2 marks you will get. Now comes the next one. Next chapter, that is the mineral nutrition. In this mineral nutrition, we have to learn only a few VSQs, not all. Okay, what define hydroponics? What is meant by hydroponics? What we have to write? The technique of growing the plants in the specific nutrient water medium without soil molecules is called as the hydroponics. That's it. Okay, growing the plants in the mineral water, that means when the elements and the enzymes are more in them and without soil, that is hydroponics. Next one. Name the essential mineral element which are responsible for playing a vital role in the photolysis of water. Photolysis of water means splitting of the water molecule. Okay. So in this, manganese, no need to write full form. Manganese, hydrogen, not hydrogen, wait. Manganese, chlorine, and calcium. These are the three elements which will help out in the photolysis of water molecules. That's it. Okay. Next one. Name the two amino acids in which sulfur is present in them. So those are the cysteine, and methionine. These are the two amino acids where sulfur is present in them. Okay. And the next one is the explain the role of pink color pigment in the root nodules of legume plants. What is it called? The pink color pigment that is present in the root nodules is the leg hemoglobin. So how to write this? Leg, you know how to write the leg. Okay, leg post hyphen hemoglobin. Leg hemoglobin is the pigment that is responsible for giving the red color. And what is it called? And this process is the and it protects the nitrogenase enzyme from oxygen. Okay, that is the one. Only you have to write this much only for the oxidation. Okay, what is which element is regarded as the 17th essential element? Name a disease caused by its deficiency. Nickel is the 17th element. Nickel is the 17th element, and the deficiency caused by this is the mouse ear in pecans. Mouse ear in pecans. Okay, finished. Now comes the next question in this. Write the balanced equation of the nitrogen fixation. Nitrogen fixation, you have to give the equation. What is the equation for this? N2 plus 8 protons plus 8 electrons plus 16 ATP gives 2 molecules of ammonia, that is NH3 plus H2 plus 16 ADPs and 16 inorganic phosphates. This is the equation what we have to say for the nitrogen equation, balanced nitrogen equation, that is N2, only one molecule of nitrogen, N2 plus 8H plus 8E plus 16 ATPs gives 2 NH3 plus H2 plus 16 ADPs plus 16 IP. Okay, inorganic phosphate. Name any, this is not necessary. Now comes the third chapter, that is the enzymes. In this, what are the important ones? What we learn in the SAQs, same thing are there in the VSAQs for this chapter. What is meant by feedback inhibition? This is the important question. What is meant by feedback inhibition? When the end products of a chain of enzymes catalyze reactions, inhibits the enzyme of the first reaction. That is called as the feedback inhibition. When the enzymes, when the enzymatic action that was there on the chain of a reaction, when the last enzyme will control the first, inhibits the first step enzymatic reactions, that is called as the feedback inhibition. Okay, then distinguish between apoenzyme and cofactor. Apoenzyme means the protein part of the holoenzyme is called as the apoenzyme. Apoenzyme means protein part of the holoenzyme is the apoenzyme. Now cofactor. Cofactor means a metal ion, otherwise otherwise organic compound that makes the bond to the enzyme is called as the cofactor. If that you write that much is sufficient. Next one. What do the 4 digits of an enzyme code indicate? Okay, enzyme code, that is the 7.1.1.2R, anything. Okay, so in this, what the enzyme code represented with the 4 digits, and in these 4 digits, the first digit represents the major class of an enzyme. This is the major class of an enzyme. Second digit indicates the class, subclass of the enzyme. Subclass of the enzyme. And the third digit indicates subclass of the enzyme. And the fourth digit indicates the number of the enzyme in the subclass. Number of the enzyme in the subclass. That is the one what they will say with the 4 codes. Okay, so the first code in this, if the first code represents the major class of the enzyme, and the second code represents the subclass, third code subclass, fourth code represents the number in the class subclass of the enzymatic classification. Okay, next comes the who proposed lock and key hypothesis? Emil Fischer. Who proposed the lock and key hypothesis and induced fit theory. In lock and key hypothesis was proposed by Emil Fischer. And induced fit theory by Daniel Koshland. Daniel E. Koshland. Okay. Now, the last one. Define Michaelis constant. What is meant by Michaelis-Menten constant, otherwise Michaelis constant? So, the substrate concentration required to cause half the minimal reaction rate. Okay. Substrate concentration, which makes the rate of reaction into the half. That is the maximum. Okay, half the maximum rate of the reaction. That is called as the Michaelis-Menten constant. With this, we have finished the 3rd chapter. That means out of these 3 chapters, 1 BSQ we can expect. Because usually they will give from that. So in the next question, photosynthesis. Where does the photolysis of water occurs? What is its significance? Where does the photolysis of water occurs? Photolysis of water occurs in the lumen of the thylakoids. In the lumen of thylakoids. Where photolysis of water occurs. In the lumen of the thylakoids. What is its significance? What it will do? It provides the H plus ions for the synthesis of NADPH2s. NADPH2s for photophosphorylation and the activation of NADP plus and activates NADP plus. Okay, this is the way by this photolysis, oxygen is going to be released into the outside atmosphere as a byproduct of the light reactions. Okay, this is the one what we have to write over here. And the next one, what is the first stable compound formed during the C3 cycle? And what is the primary acceptor of carbon dioxide in the C3 cycle? The primary acceptor of carbon dioxide in the C3 cycle is RUBP, ribulose 1,5-bis phosphate. And what is the one? What is the first stable compound that is formed? That is the 3-phosphoglyceric acid. That is PGA. Okay, like this, if you have written 2 marks, we will get. Next one, what is the primary acceptor of carbon dioxide in C4 plant? What is the first stable compound that is formed? The primary acceptor of carbon dioxide in C4 plants is the PEP, phosphoenol pyruvic acid, PEP. And the first stable compound is oxaloacetic acid, OAA. That's it. If we have written this much, we will get 2 marks completely. This one, this one. Next one is the bacteria. Now comes to the bacteria. In the bacteria also, what we have to learn, very few. Write briefly on the occurrence of microorganisms. So microorganisms, where they occur and what is the significance. Okay, and the microorganisms are the omnipresent in the vast members. Omnipresent means they will be seen everywhere. So these are the omnipresent in the many members as which are described as ubiquitous. They live in the very much diverse in the habitats. That is, everywhere they will live in a different media like wind, water, and the soil, and inside the any living organism, etc. Okay, that is the occurrence of the microorganisms. If you write like that, enough. Name the bacteria which is a common inhabitant of the human intestine and how is it useful in the biotechnology. Which is present commonly in the human intestine? E. coli. Okay, E. coli. And what is its, how is it useful in the biotechnology? It is used in the production of human insulin, that is humulin. Human insulin. To make the human insulin, we will use this E. coli plasmid DNA. Okay. And what are pleomorphic bacteria? Give example. Pleomorphic bacteria means the bacteria which can change their shape and structure according to the outside environmental conditions is called as the pleomorphic bacteria. Example, Acetobacter. Example, Acetobacter. Okay, next one. What is genophore? The chromosomal material of the main chromosomal material of the bacteria is called as the genophore. Next one. What is plasmid? What is its significance? Plasmid means a circular in the bacteria plasmid will be present now. Circular, double-stranded, extra genetic material that is present in the bacterium is called as the plasmid, and this will be they will useful in the production of the antibiotics and the used as a vector in the rDNA technology. That much we can write and we can find out. That is the plasmid and its significance. Next one, what is conjugation and who discovered this process? Conjugation means transfer of the genetic material from the one organism to the another organism through a conjugation tube is called as conjugation. This was the first discovered by Liederberg and Tatum. Liederberg and Tatum. In which organism? In E. coli organism. What is meant by conjugation? Transfer of genetic material from one organism to another organism through conjugation tube. Through conjugation tube. That is called as the conjugation. Okay, next one, transformation. Transformation, who discovered this and in which organism? Transformation means transfer of the uptake of the naked DNA from the outside atmosphere and changes the complete structure of this organism is called as the transformation. This was given by Griffith in Streptococcus pneumoniae. In Streptococcus pneumoniae. Okay, next one is the transduction. Transduction means transfer of genetic material from one organism to another organism through a viral, through a bacteriophage is called as the transduction. This was given by Liederberg and Zinder. Liederberg and Zinder. Okay, in which organism? Salmonella typhimurium. Salmonella typhimurium. Okay. Next one, viral organisms. In this, we have to learn very few VSQs. Okay. So in the virus, what is the shape of the T4 phase and what is its genetic material? The shape of T4 phase is a tadpole. T4 phase shape is tadpole. And its genetic material is DNA. Okay. Now comes the what is the shape of TMV and what is its genetic material? TMV shape is rod shape and the genetic material is RNA. This is for TMV. Okay. Next comes the what is lysozyme and what is its function. Lysozyme means a viral enzyme that can easily invade the bacterial cell wall. Okay, that was synthesized by the bacteria. And this, what is its function? It is hydrolyzing the bacterial cell wall, and the phase particles are going to be released out. That is the lysozyme enzyme. And what is a prophase? Prophase means the phase DNA which is incorporated into the bacteria during lysogenic cycle is called as the prophase. Prophase means the phase DNA. Phase DNA that is incorporated, the phase DNA which is incorporated into the bacterial DNA, incorporated into bacterial DNA during lysogenic cycle. Is called as the prophase. Okay. Next comes the principles of inheritance, 9th chapter. So in this, what we have to learn? The very first question, when a cross, what is the cross between F1 progeny and homozygous recessive parent? F1 progeny to the homozygous recessive parent, that is nothing but the test cross. And how is it useful? This is used for testing the genotype of the F1 individual. To test the genotype of F1 individual. That is the test cross. Okay. Who proposed chromosomal theory of inheritance? Sutton and Bovary. Who proposed this? Sutton and Bovary. Okay, next one. Explain the terms phenotype and the genotype. Phenotype means the physical appearance of the organism. That is called as the phenotype. Okay. And sometimes this will require the identification of the some sort of the character, that is the serological test and the respiratory quotient for the blood grouping. And the genotype means the genetic makeup of an individual is called as the genotype. If you write like this, you will get 2 marks very easily. Next one, what is point mutation? Give an example. Point mutation means when the mutation occurs at a single base pair of DNA is called as point mutation. Mutation occurs at a single base pair of DNA is called as point mutation. And the example, sickle cell anemia caused by the change in beta globin chain of from the glutamate to valine. Glutamate to valine. Okay. Now the next one. Genetic nature of the wrinkled phenotype of species. Wrinkled phenotype means that's the recessive character is going to be genotype of the wrinkled phenotype. Tall and tall. Wrinkled means smaller. Okay. Smaller. Next one. What is the phenotypic ratio? Because round and wrinkled. Round is the dominant, the capital R, and the small r is the wrinkled. Okay. What will be the phenotypic ratios of the offsprings obtained from this process? Phenotypic ratios only he is asking. The first one is capital A, small a crossed with a small a. What could be the phenotype? Tall and tall, dominant and the recessive one. Okay, 1:1 ratio. Here we will get two gametes of capital A, small a. Here we will get only one gamete of small a. This will combine with this, this will combine with this. So we will get capital A, small a. That is, one is dominant and one is recessive, 1:1 ratio. The next one is capital A, small a crossed with small a. All are dominant. All are dominant. This one, capital A crossed with small a. Then here we will get A as one gamete, here small a as one gamete, and we will get capital A, small a. That is, all are dominant. Okay, only dominant phenotypes we can see. That is 100% dominant. Otherwise, all dominant. Next one, capital A, small a crossed with small a. If that condition is there, capital A, small a crossed with capital A, small a, then we have to go with the Punnett square picture, which is very easy, beta. Capital A, small a, capital A, small a here, capital A, capital A, capital A, small a, capital A, small a, small a, small a. That means 3 are dominant, 1 is recessive. 3:1 ratio. Okay. This looks like the monohybrid cross, na 3:1 ratio. Next one, capital A, small a crossed with capital A, capital A. This one already. Okay, this we haven't done. Okay. Here, capital A will be one gamete, small a will be another gamete. Here, only capital A. That is 1:1 ratio. All are capitals, all are dominant. Capital A, capital A, capital A, small a. That means all are dominant. Okay, that is 1:0 ratio, what we can say. So, and the next one is, yeah. So see here how to write these VSAQs, because when they are asking only the phenotypic ratio in the offspring, we should not say the another one in this homozygous condition. Only one gamete we will get. We cannot get two gametes. So in some material, by mistake, they have given in this one as the 1:1 ratio, because here this is the dominant phenotype. A is the dominant. Here also one gene is A. That means all are dominant now. So we cannot get 1:1 ratio over there. 1:0 ratio only. That means otherwise what we can say, all are dominant. Otherwise we cannot say in any other words in this process. So make sure this, students, because I have seen in many materials that blunders are there in this representation part only. Only in this question, blunders are there, where they have given the 1:1 ratio, where all our dominant cases are there. So make sure that how to write it. If you don't know also, capital letter always represents for the dominant character. So like that also we can say na, when capital A, small a means all dominant characters will be expressive. That is the dominant phenotype only. Okay, right? So we will see the other VSQs, that is the 10th chapter, 2 VSQs we will get. And how to solve those, we will see tomorrow. So stay tuned to the same channel, same time for the more VSQs and the most important VSQs for the second year supply students in botany subject. Right? Thank you.